Morning, everyone. I am very pleased to introduce today's speaker, Dr. Patrick Hines, who is the founder and CEO of Functional Fluidics. Patrick Hines is an associate professor of pediatric critical care medicine at Wayne State University School of Medicine. He received his undergraduate degree from Hampton University in chemistry, his MD and PhDs from UNC Chapel Hill, and did his residency and fellowship training at Children's Hospital of Philadelphia. Patrick Hines has done quite a bit of extensive research, Ben's research actually, in hematology, particularly as it applies to sickle cell disease. The last time he spoke with us, he really enlightened us with some very interesting information, and I'm really excited to hear what he's got to speak to us about today. Dr. Hines, the floor is yours. Thank you.
Great. Thanks so much, Ned. Let me share my screen.
Yes, we can see your slides.
Okay, perfect. Just putting it on presentation mode. All right. So thanks so much for the invitation. I hope everybody can hear me okay. I'm always really excited to talk about the work that I do, just because I don't get to interact with the group as often as I'd like, and I love the opportunity to tell folks what I'm up to. And a big part of that is looking for opportunities to collaborate. I think there's some really interesting work going on in our small department from a research perspective. And so I think it's important that we all have a good idea of what strengths we have in the department, what our research interests are. And I think that will help create organic opportunities to collaborate and do some things that we're uniquely positioned to do here at Wayne State and Wayne Peds. So I'm going to talk a little bit about a big passion of mine, which is red blood cell health broadly, and sickle cell disease more narrowly, a component of that. And just along the order of background, as some folks might think, as a pediatric intensivist, how does one get into red blood cell health and sickle cell disease as a career interest? And this started back in my graduate school days at University of North Carolina, where my PhD is focused in red blood cell health and understanding the biochemistry and biology of red blood cells and how they respond to their environment, not only in sickle disease, but across many other indications where the normal functioning of red blood cells is critical. So I'm going to try to give you a little bit of background in red blood cell health, why it's important not only in sickle cell disease, but in other indications, and then to give you an update on some of the work we've been doing in sickle cell disease to really try to push the field forward in terms of how we take care of people living with disease and how we enable better research and discoveries and innovation in sickle cell disease by making these type of diagnostic and biomarker platforms available, not only here in Detroit, but nationally and internationally. So with that, I'll get started. So here are my disclosures. I have an equity interest in Functional Fluidics, which is for those who don't know, it's a spin-out company from my Wayne State-based laboratory that's based on technology that we developed in the laboratory that helps us to assess the health of red blood cells. And we apply both to development of red blood cell modifying therapies and to the care of people with red blood cell disorders around the country. So I'm going to talk to you about some of that work today will be directly related to that. learning objectives. So I want you guys to really understand the concept of red blood cell health and why it's important. Understand the role of red blood cell health in clinical disease, and understand the application of red blood cell health enabling preventative care models of sickle cell disease, especially as it relates to the really exciting time that we're in right now, where there's a lot of innovation in red blood cell modifying therapies that are going to come across your clinics. You're going to see patients not only with sickle cell disease, but with other conditions that are on some of these therapies. And I think it's important for us to have a context for what these therapies are, how they work, and how we should be thinking about them beyond what we're doing today. So this is a figure I use when I talk about the complexity of sickle cell disease. So obviously it's a single gene disorder. It impacts the hemoglobin in red blood cells, and that single gene mutation changes the chemistry of hemoglobin such that when it's deoxygenated, it has a propensity to sickle or to form these polymers. And most of us in medical school, it kind of stops there. But there's so much more biology, both in the red blood cell and directly and in the function of blood and the vascular system and systemic inflammation secondarily. So have this primary mutation in sickle cell disease that causes polymerization, but this does multiple things. Based on what we understand about the kinetic, the chemistry of sickling, the reality is that in the pathophysiology of sickle cell disease, only in extreme circumstances is actual sickling of red blood cells likely to be a big component of the pathophysiology of the disease, the actual vaso-occlusive crisis and ischemic pain and organ damage. A larger part of this pathophysiology is the fact that this abnormal hemoglobin, whether polymerized or not, creates oxidative stress inside the red cell. And that causes oxidative damage and changes to the membrane biology. These changes can induce activation of certain adhesion molecules on the surface of the red blood cell, which cause them to abnormally stick to other cells in the circulation, such as other red blood cells, white blood cells, and platelets, in addition to the vascular endothelium, which can induce systemic inflammation and can also induce the porology or blood flow in small blood vessels, because these cells actually sticking like Velcro to the vascular wall, much the way we think about white blood cells sticking and rolling. In addition to these adhesion molecules, there's changes in membrane integrity, which can shorten the lifespan of these red cells. So they're more prone to hemolysis. And with hemolysis comes the release of cell-free hemoglobin, which causes all kind of bad stuff around the body. And in the ICU, I would see the effects of this all the time, with cell-free hemoglobin sequestering nitric oxide, causing vascular reactivity, especially in the pulmonary vessels, which can lead to acute pulmonary hypertensive episode. And that's something we can see in any hemolytic anemia, not just sickle cell disease. We would see patients that would come in with hemolytic anemias from other causes, whether medication induced by external blood contacting devices. And these patients would have unexplained oxygen requirements and cardiovascular instability, which can be due in part to this nitric oxide sequestration and pulmonary vascular hypertension that's induced by this release of free hemoglobin. The other issue with the membrane affects the deformability of these red blood cells. And Dr. Ravi has done a lot of studies with a technique called ectocytometry that looks at how these cells are able to deform under normal conditions and under hypoxic conditions. And the ability of these red blood cells to deform is a big reason why we can all sit here and listen to this lecture today, because it allows these cells to pass through vessels that have a smaller diameter than the cell itself. And that's the point in which oxygen is actually exchanged with working tissue. If this deformability is not intact, then these cells are likely to get lodged into these small vessels, disrupt oxygen delivery and CO2 uptake, and can lead to a lot of the microvascular and chronic organ damage that we see in people with sickle cell disease and other red blood cell disorders. In addition, there's enzymatic metabolic defects inside these red blood cells. For example, the pyruvate kinase activation in these cells can be significantly disrupted, which helps to maintain the metabolic homeostasis within these cells. And without it, you're not able to maintain normal ion function in these membranes, which can lead to cell dehydration, which can actually promote sickling and due to hemoglobin polymerization. It can actually also reduce the membrane integrity, which can promote hemolysis and shorten lifespan of these red cells. So trying to give you guys a visual of how complex this disease is, both directly in terms of its impact on red cells, and then the secondary impact of these abnormal red blood cells, which are the largest volume of circulating cells in our blood. They're like these little inflammatory promoters that can promote vascular inflammation, white blood cell activation, platelet activation. So you've got systemic inflammatory processes, you've got thrombotic disease that you're at risk for. So you can just see how this is can be a vicious cycle and a lot of problems stemming from these abnormal red blood cells. One of the exciting things in medicine now has been the development, not only the development in terms of innovation, but the actual FDA approval of drugs that directly impact the health of red blood cells. And I use red blood cell as a umbrella term, but it can be direct red blood cell health or indirect problems caused by the abnormal function of red blood cells, as I described. Hydroxyurea was the first drug approved by the FDA for the prevention of problems in sickle cell disease, but hydroxyurea was a repurposed chemotherapeutic drug that was primarily used for leukemia, but it had secondary effect of being a ribonucleotide reductase inhibitor that increased the expression or turned off the suppression of fetal hemoglobin induction. And fetal hemoglobin, as I'm sure you guys know, has a higher affinity for oxygen. And so the thought is that it would reduce the sickling in these patients. And when taken consistently, there's many studies showing that hydroxyurea can reduce some of the clinical effects of sickle cell disease. And we believe it's through the reduction of sickling, but also we know it reduces systemic inflammation and adhesive problems in these patients. And we can see longer cell survival or reduction in hemolysis in these patients also. The problem is that there's a lot of concern about the side effects in taking chronic hydroxyurea. And there's concerns about the development of leukemias, the development of the inability of young men to have children, which is a big impediment to them being compliant with this once they reach adolescence and their early 20s. We see a big drop off in compliance with hydroxyurea. And it just doesn't work for everybody, even when they are compliant with it. So it's really important that we do a lot of work to discover other mechanisms that we can target to treat sickle cell disease. And so some other mechanisms that have been targeted have been one used by a drug called Oxbryta or voxelotor, which is a direct hemoglobin modifier. So it binds directly to intracellular hemoglobin and increases the affinity of hemoglobin for oxygen. This reduces the sickling and some of the damage that this hemoglobin causes inside of the cells, improves cell survival, and increases hemoglobin. It hasn't been firmly established that it can reduce the frequency of pain events, but this was approved by the FDA in 2019. And you can imagine the drug increases oxygen affinity. There's a lot of concern about long-term effects in that, particularly in pediatrics, where the oxygen delivery to brain tissue is a really big deal, right? So the ability to assess this directly is critical. Another mechanism that's been targeted is anti-adhesive mechanisms with a drug called Adakveo that was developed by Novartis. It's a P-selectin inhibitor, which is a adhesive protein that's located on the surface of immature red blood cells, white blood cells, and platelets. And this can facilitate binding of these cells to each other in the bloodstream and also to the vasculature. This has been shown to be a mechanism that that can drive the development of acute pain events in people with sickle cell disease. And there was a phase 2 clinical trial that showed the reduction in annualized crisis frequency in response to Adakveo, which resulted in early approval by the FDA. And I'll talk a little bit about why it's important that we don't just stop at identifying general mechanisms that can improve outcomes in sickle cell disease generally, but it's really important that we understand how this biology informs the patient-to-patient heterogeneity that should help us target the people to treat with these types of drugs. In fact, there's a good friend of me and my wife who married a gentleman that had sickle cell disease. They were living in Philadelphia at the time, and he was having issues with frequent pain episodes that weren't managed well with opioids. And so they started him on this drug. And in addition to it being efficacious in a broad, undifferentiated population of patients, there are concerns about the potential for serious adverse events because it's a monoclonal antibody, a biologic. And those of you who treat patients with biologics know that there's a subset of patients who will have bad reactions. And this was the case with Adakveo. And this gentleman, when he took Adakveo, shortly after he developed extreme pain and subsequently went on to develop multi-system organ failure, ended up in the ICU for about a month, had chronic kidney insufficiency, was on dialysis for about 4 months following that. Really horrible outcome. Fortunately, he made a complete recovery. We actually attended their wedding in Costa Rica this summer. So that was wonderful to see. But it just is a reminder of how we can't rest on our laurels with the development of these new therapies. We got to understand the biology of the people we're giving these drugs to and figure out how do we better understand and identify folks with the biology that's going to be conducive to these type of treatments. Another treatment is an L-glutamine analog called Endari, which reduces the oxidative stress inside of the membrane of these red blood cells. It's supposed to reduce systemic inflammation. But one of the unifying things around all of these drugs is that they were all approved in an environment where there's no standardized definition of red blood cell from a biomarker perspective. So these drugs, the populations were identified so based on their clinical presentation of disease. How much pain did they have? Did they respond to opiates? Had they tried hydroxyurea? Did they fail that treatment? This was the basis of starting these patients on therapy in both clinical trials and now that these drugs are FDA approved in the real world setting. And monitoring how they respond, again, is strictly based on subjective clinical response from the patients. Is your pain better? When I see you once every 3 or so months, getting that feedback from the patient. We would never do that with other chronic diseases like cardiovascular disease, where we monitor blood pressure or cholesterol. But this is exactly what we do in sickle cell disease, and this really sets the stage for an interest that I've developed since the last time that we talked in this setting, beyond just stratifying the risk of clinical outcomes, but actually personalizing and using a precision medicine approach in sickle cell disease, where we use blood-based biomarkers to define the biology of an individual patient and use that knowledge to inform how we treat these patients with this growing cohort of red cell modifying therapies. And so my theory is that if we can apply this model to red blood cell diseases like sickle cell disease, much like LDL cholesterol and cardiovascular disease or hemoglobin A1C and diabetes, we can establish patients in their baseline state that are not only at risk for having painful outcomes in a way that we can intervene early to prevent these painful outcomes as opposed to just reacting to them. We can also use a precision medicine approach to guide therapy by measuring certain critical aspects of cell function, including stickiness, mechanical fragility, and the dynamic sickling kinetics of an individual patient. And this, I believe, will allow us to better prevent and treat the complications of sickle cell disease in a preventative as opposed to a reactive way that currently is the standard of care. And hopefully that some of the data that I present to you guys will help you guys have an appreciation for that, that this is something that's indeed possible. So there a cohort of proprietary assays that we've developed in the lab that we've validated for use in people with sickle cell disease. And this is going to be the focus of the presentation, is telling you about these tests that we've developed and how we're applying them to the management of people with sickle cell disease and enabling the development of therapies for sickle cell disease. So one of them is called mechanical fragility, which is the assessment of the stability of intact red blood cells. So, I just kind of take it back to like sort of clinical place, the ICU, where when we have patients that have problems with red blood cell function, the best we can do is to measure byproducts of red blood cell destruction, cell-free hemoglobin, LDH, things like that. Well, this test allows us to assess intact red cells. And much like the tread on your tire, before you're on the side of the road, you get a sense of whether that the membrane function of that red cell is at a critical state where it's likely to hemolyze at a point where you can intervene to restore the integrity of that population of red cells. The other class of tests that we've developed is one that measures the adhesiveness or stickiness of red cells in the context of whole blood. And we look at adhesiveness to both VCAM or vascular cell adhesion molecule and P-selectin, and I'll tell you a little bit more about that. And finally, there's a class of tests that we developed called dynamic sickling, which allow us to assess the kinetics of sickling over under the conditions of tightly controlled hypoxia generation. And by assessing the kinetics of sickling, we can understand this patient's risk of sickling-induced pathology and also assess the impact of red cell modifying therapies and therapies that impact hemoglobin function. So let's talk mechanical fragility. So one of the questions we often get is, well, what's the difference between mechanical fragility and deformability? And deformability is that property that I was talking about that Dr. Ravi has done a lot of work in establishing the relationship between deformability as measured by ectocytometry, which is a specialized test used to assess deformability in red cells. Mechanical fragility is a distinct property. And the way you think about it is, if you think about the difference between a bubble and a latex glove, they're both very deformable, right? They can change their shape in response to very little pressure, but the bubble is much more fragile. It is much more prone to bursting, whereas the latex glove can withstand that force and remain and retain its integrity. Whereas the deformability, if you look at like a wine glass and a soap bubble, both are fragile objects. So a small amount of pressure can cause them to be destroyed. But the soap bubble is much more deformable than the wine glass. So that's conceptually the difference between deformability and mechanical fragility. Now, how we measure mechanical fragility it's pretty straightforward, but it's a really cool technology that we use to sort of assess it. So we take a suspension of red blood cells and subject it to mechanical shear applied by an oscillating magnet. This red blood cell suspension is a probe for hemolysis non-invasively, and this is the key. Most existing tests that assess these types of properties, you have to spin down the red blood cells and measure free hemoglobin in the supernatant. But we have a special algorithm that allows us to assess the cumulative development of release of self-free hemoglobin in response to this magnetic induced stress over time. And that allows us to calculate a mechanical fragility index for the different fractions of red blood cells in a population, from the most labile fraction to the least labile fraction, and then report that as a mechanical fragility index. And so that curve is generated. The area under that curve is essentially the mechanical fragility index. And we've established reference ranges for the MFI3 represents the most labile population within a population of red blood cells. MFI10, which is basically the accumulated hemolysis at the 10-minute mark, excuse me, looks at the total population of red blood cells. And so we have those reference ranges for the normal population and for the population of people with sickle cell disease at steady state. Now, adhesion is a metric that it's been around for a long, long time, but the problem is that because it's very complex to measure and to standardize, we've never adopted it into clinical practice. And this is really what we focused on doing in laboratory and later in functional fluidics, is establishing a scalable platform for assessing adhesion that we can standardize for both the use in clinical trials and for clinical testing in the real world. And so this is built on a well plate microfluidic platform. We've got a standard micro well plate. Underneath it is a network of microfluidic channels. And the architecture of those channels is important because you have an inlet well that you load a small volume of blood, about 15 to 30 microliters. And that blood flows through these microfluidic channels, stimulating the flow within a blood vessel. And then you capture images of the adhesion in this viewing area. Looked in cross-section, the blood that we load in the well is pressurized to drive the flow of blood across that microfluidic channel at very specific pressures that produce a very specific shear stress and pulse rate of flow. And we've done a lot of work to establish what flow conditions give us clinically meaningful adhesive results. And so essentially we just flow those cells across this surface that's functionalized with vascular cell adhesion molecule, which is an adhesion protein on blood vessels. So we sort of simulate the blood vessel surface, and P-selectin, as I mentioned, this is a major contributor to pathologic adhesive interactions. Dacthio, that drug produced by Novartis, is targeting P-selectin, and we can measure that adhesion directly. We have reference ranges for normal population and the steady state sickle cell population that we've established in longitudinal studies that we conducted right here on our sickle cell population in Detroit, that's currently used by patients around the country that are sending us samples to assess this in their patient population. This is data from our landmark study, the Ellipsis trial that we did here in Detroit, that really set the bar for how we apply these functional biomarkers for the treatment of patients with sickle cell disease. And so we looked at both fluid adhesion of whole blood, the P-selectin and VM. We established thresholds. And if a patient had adhesion indices below that threshold, then they received a score of 0. If one of their indices was above the threshold, they got a score of 1. If both were above that threshold, they got a score of 2. All right. And so at steady state, we follow these patients longitudinally and they report to us when they're having pain episodes. We could then correlate the probability of them developing a self-reported VOC event to the adhesion score that they obtained when they were at their steady state. So if that patient had a score of 0, as represented by the green, they had a 50% probability of developing a pain event beyond 100 days. So this patient when they're in their steady state, we can identify someone who has a low risk of developing a pain event in the future, much like someone with a low blood pressure, low cholesterol has a lower risk for cardiovascular disease. If they had a score of 2, in contrast, then they had a 50% probability of developing a self-reported pain event within 10 to 12 days. So this is really transformative in sickle cell disease because it allows us to identify folks that are at risk and target these therapies to the right patient at the right time. Another study that we did was establishing the role of these properties in disease by looking at this in patients who were treated with curative therapy, or in this case, hematopoietic stem cell transplantation in a patient that had a history of frequent vaso-occlusive crises. And so the red represents that critical zone above the threshold that we established in our study, and the green represents the zone in which they were at low risk for disease, for having pain events. And this is important in bone transplant and other curative therapies, gene editing, because we don't have early feedback that a patient is have a good long-term outcome. And so these patients we have to follow for, a decade or more before we can really establish that their clinical outcome is optimal. So our goal here is to really establish a much earlier signal that this patient can be predicted to have a good outcome. And this is important because it also means we can take off some of the therapies they're on so they can begin to live a normal life. So the first 2 bars represent the baseline values for these patients. And by the second baseline, they begin to get transfusion therapy to start to prepare their bone marrow for the transplant. So we see a reduction in the case of fluid adhesion of whole blood P selected. But either way, both of these values were in that critical zone, suggesting that this patient was at high risk for having pain events. 6 months after the transplant, we observe a normalization in P-selected adhesion, which continued to reduce at the year mark. In fluid adhesion to VCAM, we noticed both values were in the critical zone, and 6 months a year, they were normalized within that low risk zone, establishing the importance of these properties in maintaining normal clinical vascular function in these patients with sickle cell disease. We presented a larger version of this study, which we did in collaboration with the National Institutes of Health, where they do very sophisticated hematopoietic stem cell transplants on complex patients with sickle cell disease. And so they collaborated with us to define the normalization of red cell health in response to this type of curative therapy. So we presented this data at the American Society of Gene and Cell Therapy, where we showed, in comparison to hemoglobin assessment, which is sort of the gold standard for the response to hematopoietic stem cell transplantation, we see that we don't see a meaningful increase in hemoglobin until about month 6. Whereas when we look at VCAM and P-selected adhesion, we see a significant normalization in the functional properties of these red cells by month 3, suggesting that this may be an earlier signal of a successful transplant. It turns out that mechanical fragility does not give us that early signal. Adhesion seems to be a much earlier signal of that. And finally, I wanted to talk about with adhesion, how we are thinking about using it in the context of drug selection in patients. So I told you guys about Adakveo, the drug that was developed by Novartis. And in 2017, they started a study called SUSTAIN, where they enrolled patients based on their frequency of pain events. The main outcome was the development of pain events. And they looked at the frequency of pain events in the treated population versus the placebo population. And they showed a significant reduction in annualized crisis frequency in the treated population. The FDA required them to do a larger study in more recently that just closed at the beginning of this year. where they, again, looked at patients solely based on the frequency of pain events, not based on any kind of biology. And they measured the frequency of these pain events at a higher dose. And so it was a foregone conclusion by the community that this was going to show success, because this drug was already in the population, FDA approved, being given to patients with sickle cell disease. They did not show a reduction or any difference in treatment to placebo. And so this left the sickle cell community really scratching their heads, because they're actively prescribing this drug. Well, we established that the importance of understanding the biological type of these patients as a predictor of whether they were going to respond to therapy. In looking at a population of patients at one of our clinical send-out sites in Florida. So here, this is actually the large cohort of patients where we have an established P-selectin phenotype using our platform with up to a year before starting clinical administration of Adakveo therapy. So this isn't in a clinical trial. This is Adakveo administered in the real world. And the first thing we notice is that there's a lot of heterogeneity in these adhesive properties in these patients. And outside of sites that are actually using these tests to assess this, no one has any insight into this biology. They just give the drug to the patient, much like our friend who we talked about, who had that awful serious adverse event that put him in the ICU. And not only do we see patients that are, so this dotted line is a threshold above which we know patients have abnormally high P-selectin activity and are at risk of clinical disease. And most of these patients happen to be at higher risk, but there's a small proportion of them that weren't, that had lower values. And so that's actually the more interesting population, because when you look at that population a year post starting therapy, unlike most of the patients with elevated values who showed a significant reduction in these adhesive properties in response to Adakveo, the folks that had low values to start with actually increased their adhesion in response to P-selectin. And at year 2, we see a reduction, but it was not below where they started. So our conclusion here is that if you don't have a significant P-selectin phenotype to start with, you don't have a biologic opportunity to respond to this therapy. And in fact, you may be at increased risk for one of these serious side events, which we have multiple publications showing that with ex vivo treatment of blood samples that have low adhesion properties to begin with, we can see an increase in these adhesive properties in response to these monoclonal antibodies. So we think that this is a similar biology that's happening when this is administered to patients in the real world. And we're currently conducting a study that Novartis is sponsoring that's based at Children's Hospital, looking at these biomarkers in patients pre and post therapy across 6 consecutive infusions of the drug. And this will be the first study where we establish in a prospective study the relationship between the administration of these drugs and their baseline P-selectin activity to show whether we can actually use a precision medicine approach to identify the right population to administer this drug and establish a better method for monitoring their response to therapy. The final technology that we developed is called and this test where a lot of folks have looked at in the context of hypoxia. The difference here is that most other tests that have established the sickling use hypoxia that's induced by atmospheric oxygen or bleeding in nitrogen to reduce atmospheric reduce atmospheric oxygen. The problem here is that when you induce hypoxia in this way, you have very little control over the rate and depth of hypoxia. And in the body, hypoxia is induced in a matter of seconds to subseconds. You can't achieve that with atmospheric oxygen in an ex vivo test environment. So if we only use that environment, we aren't looking at very physiologic hypoxia rates. And 2, If you want to do this in low resource settings, either in the US or internationally, you've got to have these large nitrogen tanks to be able to do these type of tests, which is just impractical. So we developed a method of inducing hypoxia with an enzymatic approach where we can control the rate and death of hypoxia using an enzyme combination that sequesters oxygen and gives us a much tighter control where we can induce hypoxia within seconds to sub-seconds and look at kinetics under those conditions. So that's the difference in our method versus others. We can also do this with much smaller blood volumes based on that enzyme approach. And so basically we have a blood sample that has both the enzyme and the substrate. They're mixed in at this mixing block and induced and brought into this viewing area where we can observe the cells in real time and capture the development of sickling using a machine learning algorithm that assesses the change in red cell morphology from normal to sickled based on having fed it a lot of human scored cells to the point where it can do this in an automated way pretty efficiently. And so the parameters that we measure here are the morphologic point sickling, the point in which a certain percentage of the cells become sickled, the rate of sickling, the point at which 50% of the cells actually sickle, the maximum sickling, which corresponds closely to the percent of sickle hemoglobin in the sickler patient, and finally the area under the curve, which kind of integrates all of these variables into one variable. And we are able to look at a image of those cells, and machine learning algorithm can then identify cells that are of normal morphology. And as they change over time, the red box shows that the algorithm is identifying that cell as actually being sickled. It scores it as such, and it feeds that data into our algorithm. And so what that looks like in real time is we introduce the population of cells here, and we see that the system is identifying. There's some cells actually sickled to start with, which is something that we see in any blood smear that you do on a patient with sickle cell disease. There are patients who are going to be sickled as soon as you draw that blood. But when we induce hypoxia with this enzymatic approach, in real time, you can notice a point in which we get a rapid phase of sickling induction, which is happening now. Now we're having most of these cells that are actually sickling, and we reach a plateau, and that is shown graphically here on the left. And so that's basically how that happens. The cool thing is that we can show this to patients, and for patients that are on sickle disease modifying therapy that impacts the kinetics of sickling, like hydroxyurea or Oxbryta, they can actually see the impact of these drugs on their sickling kinetics in real time, which is very impactful in helping patients to become compliant with disease, because they can see this effect on their cellular biology before they can feel the clinical impact of it. And when we look at this in a similar hematopoietic stem cell transplant population, the orange bar represents the patient's pre-transplant. The blue represents the patient's post-transplant. We see that there we see a curve of a patient with a pretty typical curve of a sickle cell patient. Post-transplant, at the 6-month mark, see a significant increase in the morphologic point of sickling, reduction in the rate, and also a reduction in the maximum sickling in the area under the curve. And we just quantify those variables here in terms of the increase in the mechanical point of sickling. We never reach 50%, so there's no value there. The rate of sickling is reduced by 40%. Maximum sickling is reduced by 71%. And the area under the curve is reduced by 89%. So this is a test that we think we can apply not only in curative therapies, but in disease modifying therapies, where we can get real feedback that the patients can see and appreciate, how they're actually responding to these therapies. Beyond just the research, there's the applied science of this, which is a whole nother exercise. And how do you now take this research and make it available people who are taking care of patients in the real, once you've amassed enough data to validate how this test can be used. And so the first challenge was being CMS to approve unique billing codes for these tests so that providers at our clinical sites can actually order this test, and we can at least begin going through the process of convincing payers that there's value in obtaining this data on patients in terms of predicting their risk of having clinical outcomes and determining which therapies they should get in response to that. I'm really proud that we've been able to really build this in right here in Detroit, because clinics all over the country with the focus Florida, Mississippi, DC, Maryland, and Virginia are sending samples to us here in Detroit to be able to provide this testing on their real world clinical patients. And to date, we've done over 1,000 unique patients and done over 20,000 tests in those patients. The data that goes back to these providers looks similar to this, where we see both VCAM adhesion, P-selectin adhesion, and mechanical fragility. We don't yet have a billing code for dynamic sickling, so we're working on that. But you can see the red of the critical zone and how that patient sort of moves over time, right? And we can see this particular patient was actually started on a disease-modifying therapy around here, and we can see sort of a reduction and normalization in their adhesive properties following starting of this disease-modifying therapy. In this case, it happened to be a Dacvio. But outside of this, these patients and providers don't have any access or insight into these red cell health parameters. So this is, our approach to trying to create a platform for population scale red blood cell health assessment, so we can really become a lot smarter and use more of a precision medicine-based approach to treating these patients. These are our specific clinical pilot sites. Obviously, a lot of the initial work was done right here at the DMC and at Children's, but we've just onboarded Henry Ford, University of Miami, the Foundation for Sickle Cell Disease Research in South Florida, Children's National Medical Center in DC, and Mississippi Center for Advanced Medicine in Jackson, Mississippi. We have a number of different sites that are asking to be a part of this program, but we're limited based on our capacity. But we're hoping that we can begin to expand and make this available to more sites within the U.S. Our U.S.-based sites. So one question is how do they apply this to clinical practice? And so most of the applications are for patients that come into their outpatient clinics for baseline assessment. And either they'll have a low function phenotype or a high function phenotype or abnormal function phenotype. If their phenotype is in the normal range, if they're on disease-modifying therapy, they'll continue that therapy. They'll follow up as usual and continue to assess that patient's blood function parameters are within normal range. If that changes, that would suggest that there may be a compliance issue or a tachyphylaxis issue where the patient may no longer be responding to that same dose. And so we would, but in this case, if it's normal, we would just do standard follow-up and continue them on therapy. If that value becomes abnormal, it gives the provider the opportunity to assess compliance with this patient. Or if they're not on therapy, there's a number of these patients who are just refusing to start therapy because, in large part, in the sickle cell world, these patients are primarily African American, and there's some issues with just trust in these new innovative therapies and the research that was done behind it. So there's patients who will not consider taking these therapies, even though they're having significant clinical problems. But when you're able to show them their biology and have that conversation with them, that's a very different conversation if you're just trying to convince them to take this medication without any real evidence that it's going to be a benefit to them individually. So we've seen that this really changes the conversation around starting new therapy and compliance with therapy. The other thing that they do is they give these patients much closer follow-up. So these patients are generally home by the time we get the results to them. They'll call that patient at home, assess their clinical status. If there are any concerns, they have that patient follow up, come back into clinic. They do a repeat blood draw and assess to see if their blood function parameters are worsening. And in that case, go back and have that conversation about therapies. And in this case, they may decide if they're on therapy to add an additional therapy to try to normalize blood function, since we've got these tools that can help us do this. Finally, I want to give an update on one of the most exciting things that we've had the opportunity to do. The majority of the people around the globe that are living with sickle cell disease are not located in the US, but they're located in Saharan Africa. And so that's where the disease burden is. And as a result, for the folks that are developing these therapies and the companies that are doing these clinical trials, they're moving many of these trials to sub-Saharan Africa. But one of the problems is that they don't have the resources and the infrastructure to do this type of testing, which we've established is critical if we're going to understand who do we give these drugs to and how do we monitor it. So we have set up our first international site in Enugu in Nigeria, and that site is now under construction. This is a picture of Nnamdi Okeke, who's our head of global affairs, and he is directing the process of building out that laboratory, which is going to have the same capabilities as our laboratory here in Detroit, but it's going to be able to provide that in Nigeria, both for providers taking care of patients and for folks that are doing clinical trials. And we're already slated to start our first clinical trial by the end of the year with Pfizer on one of their second generation hemoglobin modifying drugs. And so we're really excited to be able to participate in that in Nigeria. And I'll be excited to update everybody on how that progress is going. So in closing, we really believe that red blood cell health is critical in enabling a proactive care model in sickle cell disease, not only just in predicting outcomes, but also in identifying individuals who are going to be responders to this new generation of red blood cell modifying therapies that are going to be at you guys' doorstep. You're going to see this, not only with sickle cell disease, but they're looking at this in other indications such as cardiovascular disease, diabetes, because we know that there's a lot of data showing that elevated blood sugar levels impact the function of red cell membranes in terms of deformability and adhesive properties. We've got data in neonatology where we've shown that elevated adhesive properties in moms by the third trimester can be predictive of low birth and preterm birth. And also we're looking at this in the context of people with mechanical support devices such dialysis filters, where blood contacts artificial surfaces and they can actually damage red blood cell function. So we're going to see these type of therapies not only in sickle cell disease but in other patients also. And so this is something where I hope you guys are now a little more familiar with red blood cell health as a concept, the therapies that treat it, and the importance of being able to assess and monitor red blood cell health to ensure that we're doing the right thing for our patients. So I'm going to stop there and see if there are any questions.
Dr. Hines, thank you. That was just amazing. Are there any questions from our audience?
Yeah, I've got a question, Patrick. In the last couple of minutes, you hit on what I was going to ask about, which is the global health issue. Obviously, you're a very busy guy as it is, but any plans to expand to India as well?
Yes, so India is there's a tremendous burden of sickle cell disease and other types of hemoglobinopathies and red blood cell disorders, and some of these clinical trials are being conducted in India also. So it's really about having access to a network and resources to help give you entree into these countries. Nnamdi Okeke, who was the new member of our team that set up our lab operation in Nigeria, is from Nigeria. He has great relationships with the PIs that are there in country. So the bar to getting established there was lower, but we absolutely recognize there's a need to be in India, the Middle East, Brazil, and so it's about having the resources and the bandwidth to be able to expand into those areas. So that's definitely on our radar.
But obviously through the Stem Cells and Development Journal, I've got a lot of contacts in India that are hematologists, as well as our fine people on our own staff. So if you're interested in being put in touch with any of those people, I'd be happy to do that, Patrick.
Absolutely. I would love to. Thanks so much, Graham. Alan?
Yeah, that's a great presentation. So what really struck me is the wide range of phenotypic variability in a disease that's caused by a single point mutation. And so it seems to suggest that there might be other potential genetic modifiers of the disease. And I'm guessing that the research community has pursued, this. So I was curious as to what's known, if anything, about other maybe variants in P-selectin or VCAM or other adhesion molecules. And also, if it hasn't been pursued thoroughly, it seems like you're in a perfect position to do this type of thing because you have thousands of specimens coming into your lab. And if you could consent them to do the genetic analysis, you have the phenotype data and also the response to various drugs. So it seems like a great opportunity.
Yeah, Alan, thank you for the question. And you're exactly right. There are, even when you look at the non-disease population, there's a significant amount of heterogeneity in these red blood cell functions. You've got folks that tend to be more pro-adhesive at baseline, folks who tend to be more pro-hemolytic at baseline. And this is separate from folks with sickle cell disease. We've seen this in people who seroconverted with COVID back during in COVID when we were assessing COVID serology and doing these red blood cell function testing in those populations. So we saw a significant amount of heterogeneity within the healthcare worker population that we tested. So there's a lot of genetic modifiers that impact not only the expression of specific adhesion molecules, but the biochemical second messengers that modify the adhesive affinity of those molecules at any point in time. So certain stress mediators impact certain people in different ways who are more prone to systemic inflammatory responses to different types of inflammatory mediators, such as IL-6 and IL-2 and TNF-alpha, for example. So there are many other genetic modifiers, but you're exactly right. That is the reason. This is a single gene mutation on the backdrop of a big, genetic, heterogeneous, baseline. And I think that is leading to a lot of this heterogeneity. We're really interested, Alan, in defining, much like a genetic analysis or proteomics on a population scale, defining these functional properties on a population scale, because we're seeing that's where the rubber hits the road. You can have the genetic potential, but it's not until it's manifested functionally that we're seeing your risk for certain outcomes increase. So that's our goal, is to really develop a functional nomics, to make up a word, to define these properties on a population scale.
Hey, Patrick, this is Ashok Sarnaik.
Hey, Dr. Sarnaik.
Hi, brilliant presentation as usual. My question goes to more basic and clinical level in the pathophysiology of sickling. It's a gradual process, is it not? The cell starts to sickle, and for a period of time, the sickling is reversible, and then after a certain time it becomes irreversible. My question is, your model, I mean, not model, but what you do in the lab, you have such an opportunity. I'm sure you have thought about it and actually done it also. Obviously, hydroxyurea is not will have a totally different mechanism. But are there any other therapeutic modalities that could be used early in the process before the red cells are irreversibly sickled?
That's a great question, Dr. Sarnak, and thank you for that. We tend to think about sickling as sort of the end stage of red cell pathology and sickle cell disease, and what we're learning is that before these cells actually sickle, there's polymerization is happening, and it reaches a critical level where it actually manifests as these changes in morphology. And if you look at these cells, we say sickle, but they're as many variations of this morphology as they are cells, right? So there are the cell doesn't have to be sickled morphologically at all for it to contribute to microvascular pathology. A certain amount of polymerization will lead to changes in deformability, which will impede the bulk flow of cells through small blood vessels. And so even at that point, when it's absolutely reversible, there's an opportunity for those properties to cause rheological problems. Now, to answer your question about other drug targets that can impact that, so there are drugs, if you guys remember Mike Callahan and Amar Zaidi, who are now with Agios, they're working on a drug that's a pyruvate kinase activator. And that PK activator drug increases intracellular, say, ATP, which allows normal functioning of the channels, which maintain the hydration homeostasis of these cells. And by maintaining the hydration homeostasis, you keep the concentration, the mean corpuscular hemoglobin concentration, at a normal level, and the likelihood of sickling even under hypoxic conditions. So there are other strategies that can reduce sickling while it's still reversible, other than direct hemoglobin modification with fetal hemoglobin induction or drugs like Oxbryta that directly bind to hemoglobin and increase oxygen affinity. And that's just one example.
Okay.
Did that answer your question, Dr. Sarnak?
Yes, absolutely. And as far as the India connection, please get back with me offline. I have my niece works there in cancer and other red blood cell disorders. So we may have something potentially collaboration.
Oh, great. Okay. I'll definitely follow up.
Yeah.
Any other questions?
Any other questions?
We are a little bit past time.
Thank you. Great talk.
Thank you very much,
Thank you,
Dr.
everybody, for
Hines.
your attention.
Yes. Jen will post the code for CME credit. Everybody have a great day. Thank you. Bye-bye.
All right. Take care.